Today, we're going to have an opportunity to begin the process of creating an argument so we can let Dr. Naismith know that we believe that fecal transplants should receive funding. So the first thing that we're going to do is we're going to go to Google Classroom. So if you go to Google Classroom and go to Classwork, and then we're going to click on the Microbiome Reasoning tool. When you look at it, it looks like this. One of the things about the tool is because we've already done our sense-making tracker, the evidence that we've already looked at, we've already completed that part. So we're going to use that information. We already have our claim. So if we look here at the claim, we are actually trying to figure out, because there's so much information, we're going to have 3 subclaims. So for this first subclaim, bacteria from the fecal transplant can fill up the space in the gut, limiting the food and space for invading harmful bacteria. So what we need to focus on with our reasoning tool is, how can I connect the evidence with the claim? I need to put it all together. Remember, this is the explanation part. So in our reasoning tool, it starts off and it says, this matters because. So we're going to go back to patient 23. We're going to look at his data. So I'm going to go back here into Amplify, and we're going to take a look again real quick. We talked about this yesterday. If we look at week 7, what is happening with week 7 compared to week 9? And yes, we can see, like you guys told me, the C. difficile was here, the fecal transplant occurred, and then at week 9, the C. difficile is completely gone. There is no empty space, and we actually have healthy bacteria in the gut again. So if we go back to our evidence tool, and I'm going to have you put this in your copy as well. So what you need to do, you need to have that pulled up. And right now, I'm going to have you go ahead and stop the video and type on your reasoning tool what the evidence shows us here. So the evidence shows that the fecal transplant added more healthy bacteria to the gut, which took up space and prevented C. difficile from growing. So go ahead, stop the recording, type that in your document, and once you have that typed, then hit play again. All right, so let's look at the second one. From the experiment, from the Salmonella bacteria experiment that we did. So if we go back to our sense-making tracker, so what I am doing is I am modeling this for you. You should be able to do this process, but for this first one, we're doing it together so you can see how this will work. So here is the article about Salmonella, and here was the information we wrote. When lots of helpful bacteria, like we can see here, these that are in green, when those are present in the gut, there is little space and food for the Salmonella. So if there is fewer helpful bacteria in the gut, then there is more space and food available for harmful bacteria that can cause disease. So we want to put that information into our key here, our reasoning tool. So we're going to come back to your document again on the Salmonella experiment. We're going to go ahead and type in that the experiment provides evidence that having more healthy bacteria can protect you from bad bacteria. So go ahead, stop the video, and type that in on your document. When you're finished typing that in, go ahead and hit play. All right, so you should be ready now for the third piece of evidence. This came from the article, and here it says C. difficile cannot survive if there's a lot of competition from other bacteria for food and space. So we have a theme going on. We can see that this whole thing is talking about filling up that space in the gut, and that's going back to our subclaim. So in order to help explain why this matters, if we go back to our sense-making tracker and we go back to that article on C. difficile, and I copy that information that we already put there, and we go back to our document, and we paste that in, we can see that, oh, on this side, we already have this first piece of evidence right here. So I'm going to take that out because we already have that typed underneath the evidence. So that leaves us with, after patient 23 took antibiotics, there was less helpful bacteria and a lot of empty space. So the C. difficile bacteria was able to survive in patient 23's gut microbiome. Okay, so this again is talking about that empty space and the antibiotic. And you can kind of see this works backwards. This was what we just did yesterday, day before, and the information down here was from several days ago, even last week when we first started. So here is what I would like you to do. Here we have the human microbiome article, and we have, even though it's split up a little bit here, if we look at this and we read it, even though they are tiny bacteria, even though they're tiny, bacteria are living things with the same basic needs that all living things share. The human body provides bacteria with the food and living space they need. That's what makes our bodies such a good environment for bacteria. So if we take a look here and we look at, okay, so therefore, this is what's happening. The bacteria can fill up the space in the gut. What I would like you to do is I want you to figure out, what would I put on here? Why does this matter? How does this information matter with what we're doing with this argument? So I'm going to have you pause the video and go ahead and type in your answer. What do you think you should put there to show that this information matters? How can we connect that? Okay, go ahead and stop the video and type in your answer. All right, so you should have had time to type in your answer. And let's think about this, and let's compare your answer to what we have typed. So if we look here, this is telling us, just like you learned with the amoeba sisters, and when you took your notes over the living characteristics, the characteristics that living things have, we know that living things have basic needs, and bacteria are living. Bacteria need food, they need living space. So we need to connect this, and if we take a look at it, we can see that from your human microbiome article here, you could have gone back to your sense-making tracker and put that information in as well. So what I'm going to do is I'm going to go ahead and stop this video, make sure that you have put your information in there, and we will come back and refer back to this empty space before our next activity.